Yeah, so I'm sharing my screen. Yeah. So can one of you please unmute and let me know if the screen is visible and if I'm audible?
Yes, ma'am.
Okay. Thank you. So I'd start today's session. Before starting, one notification is that this Google Meet link has a time limit. I'm not sure why because I made the time unlimited, but it has been limited to 60 minutes now. So at any point, if you get thrown out of the meeting, please rejoin using this link again. Okay. So we will rejoin using this link only, and even after 60 minutes, if all of us get thrown out of the meeting, we can always rejoin and continue the meeting till the end of the session. So today is the first live session of uh, by me of this course, Fundamentals of Protein Chemistry, with course instructor Professor Shagota from IIT Kharagpur. So basically, what we'll do is in these live sessions, we'll go over some of the previous year old assignment questions and we'll discuss the concepts behind those questions. We'll not jump from one question to another. On the contrary, we'll just take time to get through the concept of the question so that it's easier for us to solve similar type of other questions as well. Okay, so we'll discuss some of these questions like this. If you have any doubt, you can maybe unmute or write in the chat box. If I know the answer at the moment, I'll let you know, or else I'll read about it and let you know before the next class. And once the session is done, after every live session, I am making a recording of these sessions. I'll upload these live sessions on YouTube, and the link will be shared to you by the NPTEL staff. And the slides and the PPT, everything will be uploaded and will be shared, made available to you basically. So my name is Aishi, and I am a PMRF research scholar from IIT Bombay. And now we'll start the first live session of this course, Fundamentals of Protein Chemistry. So the course name is pretty self-explanatory. We'll discuss about amino acids, proteins, and the chemistry behind all of those, right? So going on to the first question for today. Which of the following options has the nitrogen heteroatom present in all the side chains of the amino acid residues? So the options are A, HKE, B, HKR, C, HDF, and D, FYW. So as you know, these are the one-letter codes of the amino acids. To answer these questions, you need to know the one-letter codes of the amino acids and also the structures of all the amino acids. So I'll wait for a few minutes. If anybody wants to answer, you can just unmute and answer and let me know the reasoning for your answer, and then we'll move ahead to the explanation part. So anybody? You can just check the amino acids corresponding to these one letter codes, check the structures, and then it becomes pretty simple. So I'll wait for 2 minutes and then we'll move ahead. In these 2 minutes, you can just check out the structures and try to answer this question. Anybody? Yes, yes. Option B, you mean HKR? Okay, okay. Great. Anybody else? Okay, so we'll discuss the this step by step. So first, if we discuss the options, H means histidine, K is lysine, E is glutamic acid, right? And then in the next option we have R, which is basically arginine. Then the next option we have D, which is aspartic acid, F, which is phenylalanine. And next question, FYW. F, I already said phenylalanine. Y is tyrosine. W is tryptophan. Right. So before going to the answer, yes, you are actually correct. It's HKR. The second option is correct. But we'll discuss about all the amino acids and the structures first before we move ahead, and why we know that B is the correct answer. Right. So if we know amino acids are the building blocks of proteins, okay, and there are four different groups that are attached to the alpha carbon: amino group, COH group, hydrogen atom, and the side chain. So this is the basic structure of an amino acid. If we see, this is the alpha. This is the carboxylic acid. This is functional group. This is the NH3 plus functional group, and we have R, which is a side chain, and we have this structure is common to all the amino acids of protein. The side chain is different for different amino acids, which makes each amino acid distinct from one another, right? And at physiological pH, which is 7.4, this COH group is dissociated, forming a negatively charged carboxylate ion, CO minus, and the amino group is protonated, forming positively charged NH3 plus. So it forms basically a Zwitter ion, right? Now, if we talk, proline is an amino acid and not exactly an amino acid because of the structure. It has a slightly different structure. Again, we'll discuss when we can see the other structures as well. Now, how do we classify the amino acids? By R group, chemical classification, nutritional classification, and metabolic classification, obviously. So, we'll be focusing mostly on the classification by R group and chemical classification. So classification by R group, then we can classify using shape, polarity, or charge. If we talk about the two categories, aliphatic and aromatic, polarity, we can talk about polar, uncharged, non-polar, and even within polar uncharged we have different options. Now, classification according to the polarity of the side chain. Now, polar amino acids will be those in which the R contains polar hydrophilic group, so can form hydrogen bond with H2. In those amino acids, R may contain OH group such as serine, threonine, tyrosine, SH group such as cysteine, and amide group as in glutamine and asparagine, and H2 group or nitrogen which acts as a base like basic amino acids which are the lysine, arginine, and histidine, and COH group which will be the acidic amino acids such as aspartic acid and glutamic acid, right? So these are the polar uncharged R groups at the moment. So if you see serine, threonine, cysteine, asparagine, glutamine, this portion is same for all. As we said, the side chain is what makes everything, all these amino acids, very distinct and different from each other. These are the positively charged R groups. If you see NH3 plus, arginine, and histidine, right? And then you have the negatively charged R groups, which is COO minus, carboxylate, glutamate, COO minus. So these will be the negatively charged R groups. Now, what are the non-polar amino acids? R is the alkyl hydrophobic group which can't enter in hydrogen bond conformation. Now, 9 amino acids are non-polar, like glycine, alanine. For glycine, the R side chain is nothing but H hydrogen only. So, the non-polar are glycine, alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, proline, and methionine. Now, these are the non-polar aliphatic amino acids. These are the one-letter code. Glycine is G, alanine is A, proline, if you see, it's a P, valine is V, leucine L, and isoleucine is I, right? Aromatic R groups. So basically, whichever will have an aromatic group attached to it, that will be an aromatic R group will be an aromatic amino acid like phenylalanine, tyrosine, and tryptophan. If you see, these are have aromatic R groups. Now, the 20 common amino acids are often referred to as with using three-letter abbreviations, right? And the structures, names, and the abbreviations for the 20 common amino acids are shown below here. So these are the non-polar aliphatic R groups, polar uncharged R groups, positively charged R groups, negatively charged R groups, and the non-polar aromatic R groups. So if you see the one parts shaded in blue are basically the side chains. As you see, the simplest one is glycine, which is nothing but H, hydrogen, and then it starts getting more and more complex from that. So in order to answer these kind of questions, you need to not only know the three-letter code of each amino acid, you need to know the one-letter code of each amino acid. You need to know what H stands for. For example, H is pretty easy because H, histidine starts with H. But then W, what does W stand for? What does F stand for? What does Y stand for? So in that case, you need to know the one letter codes of all these amino acids by heart and also the structures. In order to answer these questions, you need to know the structure of the amino acids as well. Right? So the question that we started with, the first option was HKE. H is histidine, right? So let's find histidine from here. So if you see, histidine has an imidazole ring that contains nitrogen. So obviously it has a nitrogen heteroatom in its side chain. Next we have K. K is lysine. Lysine, an epsilon amino group in its side chain, and it also has a nitrogen heteroatom. So this lysine also satisfies the condition of the question, right? Then the first option also said E, HKE. E is glutamic acid. So if you come over here, this is glutamine, but well, I'll show you the structure. In glutamic acid, you will not have nitrogen in the side chain. So in that case, that will fail to satisfy this condition, right? But in the next option, it was histidine, lysine, and arginine. Where is arginine? R is arginine. If you see, arginine has a nitrogen in guanidinium group. So this second option, it basically passes passes the condition basically because all have nitrogen in the side chains. The third option was HDF. D is aspartic acid, which again will not have any nitrogen because it will have a carboxyl group. And then we have phenylalanine with benzyl group, no nitrogen. So this option does not stand. Then we had FYW. Phenylalanine did not have any nitrogen. Tyrosine also did not have any nitrogen. Tryptophan has a nitrogen in the indole ring, but that is the only one in the group that had nitrogen. F and Y does not have. So this option also fails. So the correct answer was HKR, which is histidine, lysine, and arginine. Now, as I said, these are the one-letter codes and the three-letter codes nomenclature of all the amino acids. You need to remember this by heart, as I said. For aspartic acid, we have D, glutamic acid E. So these are a little bit difficult to remember because it does not match the first letter of the word. But still, once you start using these, then it will be easier for you to remember. Now, if we talk about nutritional classification also, it's very important. Essential amino acids are those amino acids that cannot be synthesized in the body. That means they are essential to be taken in diet. And the deficiency can affect growth, health, and protein synthesis. Whereas semi-essential amino acids are formed in the body but not in sufficient amount for body requirements, especially in children. Like arginine and histidine are known to be semi-essential, and non-essential amino acids are synthesized in the body. So it's not really absolutely essential to be taken in diet. Okay, then these are some uncommon amino acids that we non-standard amino acids, sorry, that's the right. So there are more than 700 non-standard amino acids that have been detected in living organisms, and many are metabolic intermediates, like they are in various biosynthesis pathways, like ornithine and citrulline are intermediates in the urea biosynthesis. Okay, and also there are chemical derivatives of amino acids that are have very important biological functions. Like catecholamines lack the alpha carboxylate of amino acids. If you see this, GABA, which is an aminobutyric acid, and dopamine are neurotransmitters, and histamine mediates parts of the immune response. So these are also non-standard amino acids. Now, if we talk about the functions of amino acids, apart from being the monomeric constituents of proteins and peptides, we know that the first primary structure of proteins will be amino acids, chain of amino acids. So apart from being that, some amino acids are converted to carbohydrates and are called glucogenic amino acids. And specific amino acids give rise to specialized products like tyrosine forms hormones such as thyroid hormones, T3, T4, epinephrine, norepinephrine, and a pigment called melanin. Whereas tryptophan can synthesize a vitamin called niacin. Glycine, arginine, methionine, they synthesize creatine. So glycine and cysteine help in synthesis of bile salts, whereas glutamate, cysteine, and glycine synthesize glutathione. Histidine changes to histamine on decarboxylation. Serotonin is formed from tryptophan. Glycine is used for synthesis of heme. So these are all the other functions of amino acids apart from being the monomeric constituents of proteins, peptides, polypeptides. Now, pyrimidines and purines use several amino acids for their synthesis as well, such as aspartate and glutamine for pyrimidines and glycine, aspartic acid, glutamine, and serine for purine synthesis. Now, some amino acids such as glycine and cysteine are used as deoxygens of specific substances. Then methionine acts as active methionine as adenosyl methionine or SAM, basically what we call in as an acronym form, and it transfers methyl group to various substances by transmethylation. The cysteine and methionine are sources of sulfur, right? So we have talked about proteins being functionally diverse, and there are basically polymers made from 20 standard different amino acids. They are different in characteristics and functions. So all the different proteins are made up of these amino acids, but still they are different in structure, characteristics, function, and basically all of these comes back to the primary structure of amino acids that line up in order to form the protein. Now they can perform many different functions which also depends on their structure and shape as well. They can provide structure, they can transport oxygen, direct biological reactions, control against infection, even be a source of energy. So proteins have multiple functions in multiple parts of the body, and no two proteins are similar because they a lot of factors basically determine their functions and if they are going to be working properly. So as I said, some of the class of proteins can be a structural component like collagen, which is we can see in tendons, cartilage. Keratin is in hair, skin, wool, and nails. Then it can be a contractile class of protein, like it will help your muscles move, like myosin and actin protein. They contract muscle fibers. Then it might help in transport, like carry essential substances throughout the body. Hemoglobin transpose oxygen, lipoproteins transport lipids. They might help in storage, like storing nutrients. Like casein stores protein in milk, ferritin stores iron in the spleen and liver. Then it can act as a hormone, like it regulate body metabolism and nervous system. Like insulin regulates blood glucose level. Growth hormone regulates body growth. It can function as an enzyme, like it can catalyze several biochemical reactions in the cells, being a biological catalyst. Like sucrase catalyzes the hydrolysis of sucrose, trypsin catalyzes the hydrolysis of proteins. Or it can help in protection, like it recognizes and destroys foreign substances. Immunoglobulins, basically, yeah, proteins of the immune system that stimulate immune responses. So this, as I said, this is basic. We have already discussed. Each amino acid has these three structures: a carboxyl group, an amino group, and a side chain bonded to the alpha carbon atom. Right? And these carboxyl and amino groups of different amino acids are linked together by the peptide linkage. This is the alpha carbon, side chain, carboxyl group, amino group. Now, as I said, in most body fluids, the carboxylic group and the amino group are ionized, And ionized amino acid that has both positive and negative charge is a dipolar ion called bitter ion. It will be CO minus and NH3 plus at physiological pH, and it forms as bitter ion. So we have discussed how amino acids can be classified, like with non-polar side chains, aromatic R groups, uncharged polar side chains, positively charged, and acidic side chains as well. So these are the classifications. I will not go again into this. We have discussed these as well. So, for example, non-polar side chain, the side chains that cluster in the interior of the protein due to hydrophobicity, right? And the side chain of proline and its alpha amino group form a ring structure. What is the importance of proline? Proline gives the fibrous structure of collagen and interrupts the alpha helices found in globular proteins. Then we have the aromatic R groups. Their aromatic side chains are non-polar, so that they participate in hydrophobic interactions. Tyrosine is an important in some enzymes. Most proteins absorb light at a wavelength of 280 nanometer due to aromatic groups. Okay, a property exploited by researchers in the characterization of proteins. If you are working in labs, if you have protein samples, you will be basically trying to figure out the concentration of proteins using spectrophotometer. In that case, you'll be particularly taking a particular ratio, and then you'll be checking the wavelength at 280 nanometer basically, and following Beer-Lambert's law, you'll be able to find out the concentration of the proteins. That is why it is being utilized by researchers in the characterization of proteins. Then we have uncharged polar side chains, like more hydrophilic because they form hydrogen bonds with water. It includes serine, threonine, cysteine, asparagine, glutamine, all of these. Like cysteine contains a sulfitryl group, an important component of the active site of the enzyme, many enzymes. And two cysteines can become oxidized to form a dimer, cysteine, it will be dimer, cysteine, which contains a covalent cross link called a disulfide bond. Then we know about the basic R groups. Lysine has a second positive amino group at the position on its R chain. Arginine has a positively charged guanidino group. Histidine has a positive imidazole group that facilitates the enzyme catalyzed reaction by serving as a proton acceptor donor. We have acidic side chains like aspartic glutamic acids at neutral pH. These are fully ionized. Sorry. Yes. Yeah, sure, sure, sure. Okay. Okay. Yes, yes. Okay. Yeah. So it's a good question. So when we are doing protein characterization in labs, we either use a spectrophotometer to find out the uh, concentration of proteins, or we can use various reagents as well, if you have heard like Bradford and all of those reagents. So no, not all amino acids or amino groups absorb at 280 nanometer. Only a specific group of acids show strong absorbance there. Like absorbance at 280 nanometer in proteins comes mainly from the aromatic side chains, because the conjugated pi electron systems interact strongly with UV light. So the main contributors will be obviously the aromatic amino acids like tryptophan, W, which has strongest absorbance at 280 nanometer. Then we have tyrosine, which also has a significant absorbance at 280 nanometer, and phenylalanine, which has comparatively weak absorbance. It has more around, I think, 258 to 60 nanometer, but still it contributes slightly. Histidine has very weak absorbance in UV region, not a major contributor at 280 nanometer. Like amino groups like those in lysine, arginine, histidine, they don't absorb significantly at 280. Their absorbance is mainly in the far UV, that is below 230 nanometer. So 280 nanometer absorbance is not a general amino group property. It's specifically due to aromatic amino acids. Now, there will be a question like, if only the aromatic amino acids are showing absorbance at 280 nanometer, then how are we characterizing proteins using a UV spectrophotometer? In labs, people use these to basically measure the concentration of protein, right? Either people use nanodrop spectrophotometer to measure the concentration of proteins, or they use various reagents, as I said, Bradford, Folin, and Lowry. So how do both these systems work? So that is again a very significant question. So in that case, uh, why can we measure protein concentration at 280 nanometer even though only aromatics absorb? So even though only tryptophan, tyrosine, and weak absorbance by phenylalanine is happening at 280, almost every protein has at least some aromatic residues. So the total absorbance at 280 nanometer will be proportional to that. For most proteins, the ratio of aromatics to total amino acids is relatively constant. Not perfectly, but it's almost enough for estimation. So this allows us to estimate total protein concentration from absorbance at 280. However, it's accurate if the protein sequence is known. So the extinction coefficient can be calculated. Okay, so it will be less accurate. This value will be less accurate for proteins low in tryptophan, tyrosine. Okay, and if your protein sample has any kind of contaminants like nucleic acids, suppose DNA, RNA, that can also interfere because they also absorb near 280 nanometer. Okay, so 280 nanometer works because aromatics are common and extinction coefficients are fairly predictable. Okay, now we also use Bradford or Lowry if 280 nanometer. This works, but we still use Bradford or Lowry. Why is that? Because as I said, 280 nanometer works only for relatively pure protein solutions. In many biological samples that we have in lab, we have various buffers, detergents, salts, other biomolecules, and obviously the main point is that protein aromatic in each protein, the aromatic amino acid, this content varies. So A absorbance at 280 nanometer is not really universally reliable. So we use colorimetric assays that depend on chemical reactions with peptide bonds or side chains. I'll just explain Bradford, which basically works with the Kumasi dye method. So in acidic conditions, the dye is reddish brown, which is cationic form. Upon binding proteins mostly to basic residues like arginine, lysine, histidine, and hydrophobic regions, it shifts to a blue anionic form. So the peak absorbance changes from 465 nanometer to 595 nanometer in presence of Bradford. Now this absorbance at 595 nanometer is proportional to protein concentration, which is obviously calibrated using a BSA standard curve. We use BSA to make the standard curve. Like basically we have X and Y axis. So basically we try to correlate the absorbance at 595 with the concentration using BSA, and then we figure out the unknown protein concentration from that standard curve, right? So advantage is that this is quick, sensitive, minimal interference from salts, but this process also is sensitive to detergents. Yes. So as answering your question, yes, 280 nanometer is not something that that all the amino acids do not show absorbance at 280 nanometer. It's mostly the aromatic amino acids, mostly tryptophan and tyrosine. Phenylalanine has a very weak absorbance at that. Okay, so but this process still works for purified proteins, right? When you know that there is nothing else in the system, only a proteins in solution, not detergents or rampant salts or maybe nucleic acids, so that works. But if you have a sample mixture, it's reliable to go towards these reagent based determinations like Bradford, Lowry. Okay, so that's how basically it works. Thank you. Thank you for asking this question. It's actually pretty interesting to think about when you're working in lab, because obviously it's very easy to go to a spectrophotometer and measure the concentration using that. You just add a drop in a nanodrop and nanodrop spectrophotometer. It's an instrument. Basically, you put a drop in the middle and it will give you the absorbance value. But in case of sample mixtures, it isn't very accurate, because as I said, all the proteins are not, all the amino acids are not absorbing at that, and also there will be a lot of interference from nucleic acids as well. If any of you have worked in lab, if you are isolating DNA, RNA, mostly DNA, plasmid DNA, you'll see after the isolation, you basically see the 260 by 280 ratio to see how your isolation sample is. So mostly my point is that nucleic acids will also absorb at 280 nanometer. So it's not really a very convenient system. Unless and until you have a very pure protein sample, then it probably will work, because the absorbance will be proportional to the amount of aromatic amino acids in your sample. And mostly most amino acids have a constant ratio of aromatic amino acids. So that's what the concept is all about. Anyway, moving on, we all talked about some non-standard amino acids. Some are hydroxylysine, hydroxyproline, which are found in collagen and gelatin proteins. Then we have gamma carboxyglutamic acid found in blood clotting, and methyl arginine and N-acetyl lysine are found in histone proteins associated with chromosomes. You can just remember some of the non-standard amino acids which are very important in bodily processes that should do. So there's an optical property of amino acid. So the alpha carbon of amino acid is attached to 4 different chemical groups. So it's a chiral or optically active carbon atom, right? Glycine is an exception. So what do we mean by chirality or optically active carbon atom, right? So basically the concept is uh, chirality means handedness. Basically a molecule is chiral if it cannot be superimposed on its mirror image, just like your right and left hands, right? So in chemistry, chirality usually comes from the arrangement of atoms around a central carbon. A carbon is called chiral or asymmetric if it is attached to 4 different atoms or groups, right? This arrangement makes the carbon a stereocenter, and the two possible arrangements, which will be the R and S configurations, are called the superimposable, non-superimposable mirror images, called the enantiomers. Now, what does it mean by optically active? Chiral molecules can rotate plane polarized light. So if it rotates clockwise, I think then it's called dextrorotatory or plus, and if it rotates counterclockwise, it is called lever rotatory. This optical rotation is why we call them optically active. Now in standard amino acids, except glycine. Why except glycine? Because in glycine, this R group is also H. So carbon is no longer attached to all 4 different groups. These two become same. So glycine is an exception. So except glycine, in standard amino acids, the alpha carbon is attached to H atom, carboxyl group, amino group, and side chain. So since these 4 groups are all different, alpha carbon is chiral. Glycine has 2 H atoms on alpha carbon, so it's a chiral or non-chiral, right? So yeah, so all amino acids found in proteins are of L configuration. Amino acids in aqueous solution contain weakly acidic alpha carboxyl groups and weakly basic alpha amino groups. And each of the acidic and the basic amino acids contain an ionizable group in its side chain. Thus, both free and some of the combined amino acids in peptide linkages can act in buffers. Now, formation of peptides, again, peptide is an amide bond formed between the carboxyl group of one amino acid and the amino group of the next amino acid. Okay, so the amino acid joined together in a long chain, the N terminal end to the C terminal end. This is the N terminal, C terminal. This makes a polypeptide. Okay, so example is this is glycine, alanine. If you see CONH, this is a peptide bond. This is a dipeptide, lysyl alanine. Okay. Now protein can be classified based on solubility, composition, function, shape, and size. Definitely. Uh, According to classification, proteins are divided into 3 main groups: simple, conjugated, derived. Simple proteins yield on hydrolysis and only amino acids. These proteins are further classified based on the solubility in different solvents, such as their heat coagulability. And for example, there are albumins which are readily soluble in water, dilute acids, and alkalis, and are coagulated by heat. Albumins may be precipitated out from solutions using high salt concentrations, a process which is called salting out. They are deficient in glycine. If you talk about examples, serum albumin and ovalbumin, which is basically egg white, are examples. Then there are globulins, such as they carry out synthesis, transport, metabolism. They are insoluble or sparingly soluble in water, but the solubility is greatly increased by the addition of neutral salts such as sodium chloride. These proteins are coagulated by heat. They're deficient in methionine. These are some of the examples. Histones are small and stable basic proteins. You would have heard about histone while studying about DNA packing, basically nucleic acid packing. They contain fairly large amounts of basic amino acid histidine. They're soluble in water but insoluble in ammonium hydroxide. They are not readily coagulated by heat. They occur in globin of hemoglobin and nucleoproteins. Protamines are the simplest of proteins. They are soluble in water, not coagulated by heat. They are basic in nature due to the presence of large quantities of arginine, and they are found in association with nucleic acid in the sperm cells of certain fish. And tyrosine and tryptophan are usually absent in protamines. Okay. Then we have different, I'm not going too much details, prolamines and glutalins, albuminoids. What are conjugated proteins? So conjugated proteins are simple proteins combined with some non-protein substances known as prosthetic groups. So the nature of non-protein or prosthetic groups is the basis for the subclassification of conjugated proteins. Okay. Then, for example, chromoproteins. So these are proteins containing colored prosthetic groups like hemoglobin, flavoprotein, cytochrome. Then we have lipoproteins. These are proteins conjugated with lipids such as neutral fat, phospholipids, and cholesterol. Then we have phosphoproteins. From the name, it's self-explanatory that these are proteins containing phosphoric acid. They are linked to the hydroxyl group of certain amino acid like serine in the protein casein of milk. Okay, what are derived proteins? Derived proteins are derived by partial to complete hydrolysis from the simple or conjugated proteins by the action of acids, alkalis, or enzymes. So they can be primary derived or secondary derived. So primary derived proteins are formed by process causing only slight changes in the protein molecule and its properties. There is little or no hydrolytic cleavage of peptide bonds. Okay, secondary derived proteins are formed in the progressive hydrolytic cleavage of the peptide bonds of protein molecule. They are roughly grouped into proteoses, peptones, and peptides. Okay. Now, according to function, proteins can be subcategorized into enzymes. We know that the most striking characteristic feature of these proteins is that their ability to function within the living cells as biocatalysts. Okay, so they are called enzymes. They basically can enhance the reaction rates almost a million fold. Then we have regulatory proteins like hormones, which are basically found in relatively lower concentration in the animal kingdom, but they play a very important regulatory role in maintaining order. Contractile proteins, secretory proteins, all of these. We have protective proteins, which are antibodies. They have protective defense function. These proteins combined with foreign protein and other substances and fight against certain diseases. Then we have storage proteins, which are a major class of proteins which has the function of storing amino acids and nutrients and has building blocks for growing embryo. So transport proteins, obviously they help in transporting, binding and transporting specific types of molecules through blood. Hemoglobin is conjugated protein composed of colorless basic protein, the globin and the ferroprotoporphyrin or heme. So it can bind with oxygen and transport through blood to various tissues. Myoglobin, a related protein, transports oxygen in muscle. So lipids bind to serum proteins like albumin and transported to as lipoproteins from in the blood. We will not go into too much details. We have read about all of these. Polar amino acids, semi-essential amino acids, as I said. Essential means they are essential for your diet. Semi-essential means they are formed in your body but still not in sufficient amount. And non-essential means they are formed in the body in amount enough for adults and children. Okay, we have glucogenic amino acids which basically give glucose. They include the rest of amino acids. So these amino acids by catabolism yields products that enter in glycogen and glucose formation. So we have the ketogenic amino acids which give ketone bodies like lysine and leucine are pure ketogenic. Then we have mixed ketogenic and glucogenic, which give both ketone bodies and glucose, like isoleucine, phenylalanine, tyrosine, and tryptophan. Now, what is the amphoteric property of amino acids? That is, they have both basic and acidic groups, so can act as a base or acid. So, neutral amino acids exist in aqueous solutions as bitter ion. So, what is the isoelectric pH? It's the pH at which the zwitterion is formed. Okay, at which the basic net charge will be basically zero. So what can be the chemical properties of amino acids? There can be reactions due to either the COH group or the NH2 group. So they can be salt formation with alkalis, ester formation with alcohols, amide formation with amines, and decarboxylation. For NH2 group, they can be deamination and reaction with anhydrine reagent. Anhydrine reagent reacts with amino group of amino acid yielding blue colored product. So the intensity of the blue color basically indicates the quantity of amino acid. It's a very typical test done in laboratories. Okay. Uh, there are various other reactions that can also happen due to the side chains. So yeah, so what is, we talked about peptide bond formation. Alpha carboxyl group of one amino acid with side chain R1 forms a covalent peptide bond with alpha amino group of another amino acid with the side chain R2 by removal of a molecule of water, and the result will be a dipeptide. By the same way, the dipeptide can then form a second peptide bond with the third amino acid to give a tripeptide. So the repetition of this process generates a polypeptide or protein of specific amino acid sequence. This is the N terminal and the C terminal. Okay, the left side is the free amino group of the first amino acid that entered in the chain formation, that is the N terminus, and the right side the free COH group of the last amino acid termed as the C terminus. Okay, so yeah, there are 4 levels of protein structure. We'll come at this later in the next questions. We'll move ahead. We'll discuss the next questions. Wait, we'll not go into these details. We have talked about optical property. We have talked about these. Yeah. Move on to the next question. Yeah. We have talked about optical property as well. Okay. So coming back to the question that we started with, which of the following options has the nitrogen heteroatom present in all the side chains of the amino acid residues? So the answer will be HKR. We have discussed because it will be histidine, lysine, and arginine, right? We discussed the other, in all the other options there was at least one amino acid which did not satisfy the condition. So HKR is the correct option. So anybody has any question? I'll wait for 1 minute and then we'll move on to the next question for today. Anybody, any question? Okay, then we'll move ahead to the next question for today. Identify the regions marked X, Y, and Z in the Ramachandran plot shown. Okay. So options are A, X right-handed alpha helix, Y left-handed alpha helix, Z beta sheet, B, X left-handed alpha helix, Y right-handed alpha helix, Z beta sheet, C, X beta sheet, Y left-handed alpha helix, Z right-handed alpha helix, and D beta sheet, Y right-handed alpha helix, Z left-handed alpha helix. So for this we need to know the Ramachandran plot exactly, and then only we can answer. So we can explain the Ramachandran plot in detail. But I'll wait for 2 minutes. If anybody wants to answer the question, try. If you want to try it, please go ahead. I'll wait for 2-3 minutes. Anybody? So for this, we need to have a working knowledge of the Ramachandran plot. So I'll explain that to you. If anybody wants to try before that. So, okay, so what the Ramachandran plot basically is, it's a map of allowed backbone conformations of a polypeptide, right? So it plots the possible rotation angles around two backbone bonds. Phi, rotation between the N C alpha bond, and psi, which is rotation around the C alpha C bond, right? So this answer will be C. X will be beta sheet. Y is left-handed alpha helix. Z is right-handed alpha helix. But anyway, moving on, we'll discuss. Then we'll come to the answer. So if you talk about protein backbone, this is a biochemical view. This is a basic repeating unit is NH, C alpha H, C, and O. So we can also look at the repeating units from C alpha to C alpha, right? So what are the interesting properties? Bond lengths almost equal in all groups in all proteins, and bond angles almost equal in all groups in all proteins. So a C alpha atom belongs to two units, all atoms in a unit coplanar, and all preferable when describing structural properties, right? So geometric and structural view, polypeptide chains divided into peptide units like C alpha atom and carboxyl group of residue I, amino group and C alpha atom of residue I plus 1 are rigid group. Peptide units are rigid groups and they have rotation on bonds. CN is prevented by energy barrier. So peptide units are joined by covalent bonds between C alpha atoms, right? So thus peptides can rotate along two bonds, N C alpha okay, which is the phi rotation, and C alpha C, which is the psi rotation. Okay, if you can see over here, I'll just phi and the psi rotation. Now, two dihedral angles for each unit phi or psi, 2 degrees of freedom per unit, and it determine the conformation of the backbone as well. So, if we talk about dihedral angles and regular structures, so repeating values of phi and psi along the main chain result in regular structure. So, if the repeating values of phi is minus 57 degree and psi minus 47, it can give rise to a right-handed helical fold, alpha helix. Okay, repeated values of this 110, 140 and psi 110 plus 110, 135 can give subchains with conformations that allow interactions between nearby parallel segments like beta sheet, right? And most combinations of phi and psi are not allowed. Allowed conformations plotted as 2D chart. It's a Ramachandran plot. Now, what is the secondary structure? Defined by basically patterns of hydrogen bonds between backbone amide groups. So side chain, main chain, and side chains. Yes. Which one? The back? Okay. Okay. Okay, so basically Ramachandran plot is a map of allowed backbone conformation of a polypeptide, right? So it plots the possible rotation angles around two backbone bonds, like phi rotation, which is the N C alpha bond, and the psi rotation, which is the C alpha and C bond. Now each amino acid residue in a protein has certain phi psi angles that determine its conformation. Now, why only certain angles are allowed? Because atoms in the backbone have steric hindrance, right? They cannot overlap due to van der Waals radii. And most combinations of phi and psi cause atoms to clash. So that's why those are called the forbidden regions. Only certain combinations avoid clashes, so those are the allowed regions. Okay, so this is a typical Ramachandran plot. If you look at this, the phi on the x-axis and the psi on the y-axis. The top left quadrant, which is uh, phi will be from minus, I think minus 60 around, and psi will be till minus 40. I think these regions, that will be a right-handed alpha helix, that is common in proteins. Which one? This one. Okay, this is the right-handed alpha helix. So these are, you have to remember all of these, that these are for, there are some forbidden regions, there are some allowed conformations, but these exactly where in the Ramachandran plot what will fall, the exact values you might have to remember. Okay, now top left quadrant, more negative, these will be the beta sheets, right? So psi will be 5 will be, I think, minus even 120 and all, and psi will be positive 120. So these beta sheets can be parallel or anti-parallel. Now top right quadrant will be your left-handed alpha helix here. Okay, they are rare and more accessible to, like for example, glycine, and other small regions would be various different turns and loops. Okay, now glycine, why I say glycine is a special case, because glycine has no side chain, right? It has just hydrogen, so it's very flexible, allowed in many more regions, dense scattered. So glycine will be a dense scattered on the plot because it has two hydrogen atoms, so that's why the steric hindrance across is not that much, right? While proline side chains bonds back to N. So it has a restricted rotation, very limited range of phi values, which is why proline you will see in very limited regions, and then there are pre-proline residues as well. So why it's important? Because it validates protein structure from X-ray and NMR. Residues outside the allowed regions may indicate errors, unless obviously there are special cases like glycine, and it also helps predict secondary structure. Now, if there's a cluster of points in the alpha region, we'll assume it's an alpha helix. Cluster in beta region, there will be a beta sheet. right? So in short, the Ramachandran plot is a static map of allowable phi and psi combinations shaped by atomic collisions and is directly linked to secondary structures. Okay. So as I said, these are the secondary structures, mostly alpha beta sheets. There's obviously a lot of uh, scattered on the plot for turns and loops, but we'll mainly be focusing on the alpha helices and beta sheets. For example, if we focus mostly on alpha helix, it's a right-handed coiled formation, right? So the backbone NH group, the I plus 4 forms hydrogen bonding with backbone CO group I. So the first amino acid with the fourth amino acid will be forming a hydrogen group, and per turn there are 3.6 residues per turn, right? So there will be variations with chain more loosely or more tightly coiled are also possible, but not often. It's mostly 3.6 residues per turn. Okay, and the backbone dihedral angles are around -60 -45, and the sum of the phi and chi angles will be around 105 degree. Okay, so all the H bonds point in the same direction. It is aligned along a helical axis. So this is what alpha helix is all about. Now, if you talk about beta sheets, you have to see the image, but you have to also imagine in a 3D system, right? In beta sheets, it's a combination of several regions of the chain and beta strands, like parallel, all amino acids go in the same direction, evenly spaced H bonds. Anti-parallel will be amino acids in successive strands, alternate directions, right? And they can be mixed beta sheets also with parallel and anti-parallel strands. So they have twisted strands, right-handed twist, right? This is how it will be look in a cartoon ribbon system. Now, proteins are basically built from the primary structure, which is the chain of amino acids that form the protein. Then we have the secondary structure, right? So the structure is basically starts building from the secondary structure. They are connected by various loop regions, various lengths, irregular shape, and they're at the surface of the protein. They can reach in charged and polar residues, so they are easier to predict from the only the amino acid chain. You can predict the structure of the and shape of the protein. How fascinating is that? And if there are in homologous proteins, if there are any kind of mutations such as insertion or deletions, you can also predict the change in structure because of that. So there can be structural motifs from the secondary structures. We can actually predict the kind of motifs they'll form, like for example alpha helices and beta sheets in a motif. So they'll be, if they are adjacent in three-dimensional structure, they can be connected by loop regions. Okay, and the motifs and the secondary structures combined can be called domains. Now if you ask me what is the difference between domains and motifs in protein, so basically what happens in uh, for example, in motif's definition is it's a short recurring pattern in a protein that is associated with a specific structural function. Like it often has a specific arrangement of secondary structures like alpha helices or beta sheets in a characteristic way. They're too small to function independently, right? So they usually contribute to the function of a domain or a protein. It is recognized as a structural or sequence pattern. For example, helix turn helix motif, common in DNA binding proteins. Zinc finger motif, that binds the zinc ion to stabilize structure for DNA RNA binding. Then we have beta alpha beta motif, that is a common fold in enzymes. Then if we talk about domains, domains are larger, stable, independently folding structural unit of a protein that can often function on its own. They're usually 40 to even 200, 300 amino acids long, and they can exist and function independently when separated from the rest of the protein. So a single protein can have multiple domains, like it's a modular design. For example, if you have heard about the SH2 domain, which binds phosphotyrosine residues in a signaling proteins. If you go through the cell signaling chapter, you will find various proteins with SH2 domains, right? They act as like adapters. Okay, then we have kinase domains, catalyzes phosphorylation. Then we have immunoglobulin domains, that are structural scaffold in antibodies. This is the basic. A motif is like a phrase in a sentence, like it has meaning, but it's not fully independent. But domain is like a full paragraph. it has a complete unit of meaning that can stand on its own. That is the difference between domains and motifs. Then we have tertiary structure of protein, which is basically the arrangement of secondary structures, structural domains, how the alpha helices, beta sheets, or loops, turns, how they're arranged in the 3D structure in space, how they're arranged. Then we have the quaternary structure, which is more than one polypeptide or protein subunits are folded together. Okay, so the native conformation can be direct consequence of primary structure, chemical environment like water based, oily, or whatever. So far, no reliable computational method exists to predict the native structure from the amino acid sequence. So there are computational methods, but only that cannot. There are systems such as AlphaFold and all of that. People are doing that, but then you still those are still predictions. You need to verify that using purifying the protein and using process like X-ray crystallography or cryo-EM, which is even more popular nowadays. Okay. Those structural classes, protein structures can be four classes: alpha domains, like code built only from alpha helices; beta domains, code built up from usually two antiparallel beta sheets; alpha beta domains, like they can be beta alpha beta motifs, like parallel beta sheets surrounded by alpha helices. Then we have alpha plus beta domains, like few cases, anti-parallel beta sheet packed against alpha helices. Okay, so for example, in properties of the peptide bond, in extra studies of crystalline peptides, Linus Pauling and Robert Corey found that the CN bond in the peptide link is intermediate in length between typical CN single bond and a CN double bond. So the bond that they saw is intermediate. It's not exactly a single bond, not exactly a double bond. So they also found that the peptide bond is planar. All four atoms attached to the CN group are located in the same plane, and that the two alpha carbon atoms attached to the CN are always trans to each other on opposite sides of the peptide bond. So what does the length of CN bond in the peptide linkage indicate about its strength, bond order, etc.? So double and triple bonds are shorter and stronger than single bonds. Because the length of a peptide bond more closely resembles that of a double bond, it suggests that a peptide bond has some characteristics of a double bond as well. Thus, a peptide bond is predicted to be stronger than a typical single bond, right? And the conclusion that a peptide bond resembles a double bond is further supported by the fact that the two alpha carbon atoms attached to the CN peptide bond are always trans to one another. So rotation about this bond is apparently impossible, right? So these are some of the in one of the books, I think, from Leninger. So for example, this, if you can see, interpreting Ramachandran plot, examine the two proteins labeled A and B below. Which of the two Ramachandran plots labeled C and D is more likely to be derived from which protein and why? So the proteins shown in this panel, A and B, are dominated by beta. If you see here, these are mostly beta sheets. There is only this alpha helix, this is alpha helix, but otherwise mostly beta sheets. Whereas here it is mostly alpha helices in this protein. It's dominated by alpha structure. So based on the phi and psi angles, beta conformation will occur in the upper left quadrant, right, of Ramachandran plot. Whereas alpha helical structure lies in the lower left quadrant. So there is more likely that the Ramachandran plot shown in panel C belongs to the protein in panel A. Okay, because it is mostly beta sheets. If you see all these dots, so this is belongs to the, and the plot in panel D belongs to the protein in panel B. Because here you have alpha helices as well. The beta sheet portion is not that crowded, and that makes sense because here we do not have such as much beta sheets, right? So these are some of the, here it is, so a simplified way to understand Ramachandran plot. So if you see over here, the partial double bond character keeps the peptide bond in a rigid planar configuration. Hence, for a pair of amino acids linked by a peptide bond, 6 atoms lie in the same plane. Okay. So what are torsion angles or dihedral angles, and are defined by 4 points in space. So if a system of 4 atoms, if you see the here A, B, C, D is projected onto a plane normal to BC, The angle between the projection AB and the projection of CD is described as the torsion angle between BC, and this angle may also be described as the angle between the plane containing atoms A, B, and C in the plane containing B, C, and D. So it's easier to visualize. I think you have to, apart from seeing the image, you also have to visualize in your mind how it will look from look in space. So peptide linkage has partial double bond character, as we learned before, right? So rotation above this bond is restricted. So two possible configurations, cis and trans. If you see, this is the cis configuration and this is the trans configuration, are observed for a planar peptide bond in polypeptide. So in the cis configuration, successive alpha carbon atoms are on the same side of the peptide bond. See, these alpha carbons, carbon alpha, are on the same side. However, in the trans configuration, the two successive alpha carbon atoms are on opposite sides of the peptide bond. The restricted rotation about C N bond can be specified by torsion angle omega, right? So in the trans configuration, omega is 180 degree, plus in the cis configuration, omega is 0 degree. So what is this torsion angle omega, right? So this is how uh, so this peptide linkage has a partial level, basically how we started the conversation. So what is this omega or torsion angle, which basically changes in the cis and the trans configuration? So moving on to the next slide. Okay, one second. This one, right? One second. Yeah. So trans configuration is favored by a ratio of approximately 1000:1 over the cis configuration. Okay, and this preference for trans over cis can be explained by the fact that steric clashes between groups attached to the alpha carbon atoms hinder the formation of cis configuration, but do not arise in the trans configuration. However, the steric interference is reduced in peptide bonds to proline residues. Therefore, some peptides containing successive proline residues are known to prefer cis configuration. If you see, this is the torsion angle omega, and this is the trans configuration, which is much easier to find in nature. Now the bonds between the nitrogen of amino group and the alpha carbon atom, and between the alpha carbon atom and the one of the carbonyl group, are pure single bonds. So the two adjacent rigid peptide units can rotate about these bonds, acquiring various orientations. The rotations about these bonds can be specified by the torsion angles phi and psi, what we were discussing, right? About the different phi and psi, basically that makes up the Ramachandran plot. So theoretically, phi and psi can have any value between 180 plus 180 and minus 180, right? However, not all combinations are possible in reality due to the physical clashes of atoms in 3D space. Atoms take up space and two atoms cannot occupy the same space at the same time. So these physical clashes are called steric interference. So most values of phi and psi are therefore not allowed due to steric interference between non-bonded atoms. So the permitted values for phi and psi were first determined by G. N. Ramachandran, and these permitted values can be visualized on a two-dimensional plot called the Ramachandran plot. Okay, what we are discussing, plotted between the phi and the psi on the x and the y axis respectively. So polypeptide conformations are defined by the values of pi and psi. Most values of pi and psi are not allowed due to steric interference between non-bonded atoms. Hence, most areas of the Ramachandran plot represent sterically disallowed conformations of polypeptide chain because of steric collisions between the side chains and the main chain. Okay, we see over here in figure 6, the white areas correspond to sterically disallowed conformations in which any non-bonding at interatomic distance is less than its corresponding van der Waals radii, and these regions are sterically disallowed for all amino acids except glycine. It is unique as it lacks a side chain. As I said, glycine is a special case because both there are two hydrogen atoms in glycine. Now the dark blue regions are called the allowed regions, correspond to conformations where there are no steric interferences. Most of phi and psi values of a polypeptide chain fall within these allowed regions of the Ramachandran plot. There are, however, some notable exceptions like glycine. The amino acid with the smallest side chain is much less sterically restricted than the other amino acid residues. Hence, its allowed range of phi and psi covers a larger area of the Ramachandran plot. If you see, one second. This is glycine. See how the dark blue region is so much more because of the two hydrogen atoms. There is less sterical hindrance in most of the conformations. Okay, and glycine residues, polypeptide chains often assume conformations that are sterically disallowed to other residues. So the cyclic side chain of proline limits its range of phi values to almost around -60, making it most conformationally restricted amino acid residue. Okay, the light blue regions correspond to conformations having outer limit van der Waals distances, that is, the atoms are allowed to come a little closer together. Now, since the D and the L form of amino acids have the side chain oriented differently with respect to the CO group, hence they have a different allowed phi and psi angles. So, if polypeptides built from D amino acids, they have a different phi and psi angles than those that are exclusively made of L amino acids. For example, a beta sheet made up of L amino acids will occupy upper left quadrant, whereas beta sheet made up of same amino acids of D enantiomer form would occupy the lower right quadrant. Okay, so these are the basically the glycine residues, how it looks. So all of that. So this is now, how can you predict structure from Ramachandran plot? So every amino acid residue in a polypeptide can have specific set of phi and psi values, right? Angles. Therefore, each residue can be represented as a point on the Ramachandran plot with corresponding phi and psi angles, respectively coordinates, x and y coordinates, right? So, polypeptides, when adopting secondary structures, rotate at specific torsional angles each time so as to form regular repetitive structures such as the alpha helix and the beta sheet. Therefore, on plotting these torsional angles to Ramachandran plot, we obtain a very restricted area on the plot which can be used to identify and check the secondary structure in a given polypeptide. So, the backbone torsion angles for right-handed alpha helix are approximately phi minus 57 and psi minus 47 and therefore occupy small area on lower left quadrant. Beta sheet is made up of mostly fully extended strands with alpha and with phi and psi angles falling in the upper left quadrant of the Ramachandran plot. If you see over here, the top portion is mostly anti-parallel, then parallel, then right-handed alpha helix, left-handed alpha helix. Okay, so these are basically what is the table contains the idealized phi and psi angles for common secondary structures in proteins. You can try to remember the most important ones like alpha helix and beta sheets portions. Okay, moving on. Yeah, so we'll not go into too much detail. This is the, yeah, so this is how it looks. This was the diagram from the question. The topmost is the beta sheet, then you have the right-handed alpha helix and the left-handed alpha helix on this side. Okay, so this is how it looks. So the answer will be, this will be the beta sheet. X. Z will be the right-handed alpha helix and Z will be the and Y will be the left-handed alpha helix. Okay, so that is the correct answer. Option C. So anybody has any questions regarding this? We can discuss, or I'll wait for a few minutes and then we'll move on to the next question for today. Anybody, any questions? I'll wait for a few more minutes and then we'll move on to the next question for today. Okay, so moving on to the next question for today. Chaperones are examples of ribonucleases. True or false? Anybody? So, what are chaperones in normal English? What do these things mean? And what are molecular chaperones? I'll wait for a few minutes. You can try answering the question, and after that, I'll move on to the discussion. Anybody? You can try also, you can check and try. Okay, so starting off, if we talk what is the meaning of chaperone, so a chaperone in everyday life is someone who guides, supervises, or protects another person, right? Often to help them behave or reach a destination safely. Okay, that is the meaning of chaperone in normal English. Now, what do we mean by chaperone in biology? So, the idea is very similar. Like proteins or other molecules in the cell, sometimes need help to fold correctly or reach the right place. So, molecular chaperones are special helper proteins that guide other proteins during folding or assembly to make sure they don't misfold or clump together. So what do they do? Newly made proteins are likely fresh sewn like clothes. They can get wrinkled, tangled, or stick to the wrong things, right? So chaperones act like, you know, basically helpers or basically guides that help them fold into the correct 3D shape. It does not stick to the wrong partners. Like it does not undergo aggregation, and it can refold after stress. So they do not determine the final structure, they just assist the protein in finding its correct shape. Okay, this answer is false. These are not examples of ribonucleases, rather, these are helper proteins, for example. So we will go about this discussion. So we'll not go into very much detail if we will move directly to protein misfolding and chaperones, right? Now proteins can fold and they do so fairly fast. Okay. And protein funnel is a good model, like extending beyond nearest neighbor um, interaction. Okay. So what do we mean by the protein folding model? Okay. We'll discuss about that as well. So if we talk about the protein folding energy landscape, proteins fold because the native 3D structure corresponds to the lowest free energy state under physiological conditions. So the process is guided by the energy landscape, which maps all possible conformations of the proteins against the free energies. Okay, so in most cases they don't need help, they can do on its own. But in complicated cases like big proteins, very crowded conditions such as in a cell, proteins get help with helper proteins called chaperones. So this is the protein folding the energy landscape theory. Okay, so as I said, for proteins fold because the native 3D structure corresponds to the lowest free energy state under physiological conditions, and the folding process is guided by the energy landscape, which maps all the possible conformations of the protein and their free energies. Okay, so basically think of it as a map showing which conformations are stable and which are unstable, and how a protein moves from unfolded to folded states. So the classic visualization is the folding funnel model, right? So the top of the funnel, this portion is the unfolded. It represents the unfolded state. So there are many possible conformations. It has a very high entropy, right? High free energy due to solvent exposure of hydrophobic residues, unoptimized interactions, and all of that. Now the sides of the funnel, as the protein starts folding, it samples fewer conformations, right? The entropy starts decreasing. Energetically favorable interactions, hydrophobic core formation, hydrogen bonding, van der Waals packing, they drive the protein downward in free energy. And then we have the bottom of the funnel. It represents the native state, low free energy, high stability. It's unique or very limited conformation that is functional. Okay, so what is the thermodynamics of folding? For example, the free energy change or the delta G. The folding is usually spontaneous, like delta G will be lesser than 0. Now we have to talk about enthalpy, delta H, stabilizing interactions like hydrogen bonds, salt bridges, van der Waals forces, all of these release energy. And if we talk about entropy, delta S, loss of conformational entropy is compensated by increased entropy of water due to hydrophobic effect. So the balance is that the folding occurs because favorable enthalpic and solvent entropy contributions overcome the conformational entropy loss. Okay, now proteins, there are a lot of conformations that a protein can fold into, right? But proteins do not explore every conformation randomly. Okay, there's this thing called the Levinthal's paradox. What they do is that the local secondary structures form rapidly, like the alpha helices, beta sheets, and these nucleate intermediate structures, they form the molten globule states, right? And finally, small arrangements produce the fully folded native state. Okay, some proteins may fold directly to native state to state folding or pass to intermediates like the multi-state folding. Okay, now why do proteins get misfolded? Most proteins can spontaneously refold, like primary sequence determines tertiary. Some proteins do not. Like if you boil an egg and bring temperature back down and it won't reform, right? Albumin grows from clear to milky white. Commonly, the hydrophobic residues get exposed. So when concentration of protein is high, they can fold up with other proteins instead of with itself and remain unfolded and aggregated. So there are a wide variety of such proteins, similar structure and bad outcomes. So there's the amyloid fiber and plaques. They can lead to the mad cow disease, the Alzheimer's, disease, Parkinson disease, all kinds of neurodegenerative disease, maybe some forms of diabetes as well. Now, protein folding in the cell. Most proteins probably go through several stages on their way to a stable structure, as I said. Some need help, particularly in a crowded environment of cell. So, chaperones are nothing but protein molecules that assist the proper folding of other proteins. Okay, they're often are heat shock proteins because they help cell with elevated temperatures, which tend to cause proteins to misfold. Also involved in newly synthesized proteins where a lot of hydrophobic groups haven't yet folded up properly. Some chaperone systems like the chaperonins work as foldases. So they support the folding of proteins in an ATP dependent manner. For example, the GROW-ES system. You can read about these systems in detail in books and online. Other chaperones work as holdases. So they bind folding intermediates to prevent their aggregation. In that case, no ATP is required. Neurodegenerative diseases such as Alzheimer's, Parkinson's, and mad cow disease are all associated with misfolded proteins. Okay. So when intermolecular contacts are significant, they need to modify the energy funnel, as you were discussing. Assume that the 50% of the contacts are formed, then can either fold normally in dilute conditions or it can react with another partially unfolded protein and fall down to the aggregation tunnel funnel. Okay. So lysozyme is a well-studied example of misfolding. Lysozyme is abundant in a number of secretions such as tears, saliva, human milk, mucus, also in egg white. They damage the bacterial cell walls through catalyzing. It can form amyloid fibers. So there are basically lysozyme has been studied a lot as a subject for misfolding. Now, if we come to prions, they are what are prions? They are a really hot topic now. Various labs in the country and abroad are working on this. So prions are infectious agents that can be transferred which are devoid of nucleic acids. Okay, so basically prions have been added to the list of infectious agents like bacteria, fungi, and parasites. However, prions exist normally as innocuous cellular proteins, generally called PRP. They are no pathogens, they are basically proteins. However, prions possess an innate capacity to convert their structures into highly stable conformations that ultimately result in the formation of harmful particles. So, the causative agents of several deadly brain diseases of the dementia type in humans and animals. This is called PRP-SC, where SC stands for scrapie, basically. The original prion disease where it was discovered in sheep. So PRPSC is very stable. Resistance to degradation where number of proteases, heating, chemical denaturation doesn't work. So it gets misfolded, it gets aggregated, and then it's very stable. So prion diseases may be inherited, laterally transmitted, or occur spontaneously. And regions within diseased brains have a characteristic porous and spongy appearance, evidence of extensive nerve cell death and affected individuals exhibit neurological symptoms, including impaired muscle control, loss of mental acuity, memory loss, insomnia, etc. So, a prion in the scrapie form, like PRPSC, is an infectious agent composed of protein in a misfolded form. This is the central idea of prion hypothesis. This would be in contrast to all other known infectious agents like viruses, bacteria, fungi, parasites, etc., all of which contain nucleic acids. The PRPSC can catalyze PRP to form amyloid fibrils. What happens is that the misfolded aggregated protein PRPSC then takes control of the normally folded proper protein and then makes catalyzes that also to form amyloid fibrils. So basically the number and the amount of misfolded aggregated proteins keeps increasing. So prions are not considered living organisms but are misfolded proteins molecules which may propagate by transmitting a misfolded protein state, right? So if a prion enters a healthy organism, it induces existing properly folded proteins to convert into disease associated misfolded prion form, and the prion acts as a template to guide the misfolding of more proteins into prion form. So these newly formed proteins can then go to convert more proteins themselves, and this triggers a chain reaction that produces large amounts of the prion form. So all known prions induce the formation of an amyloid fold in which the protein polymerizes into an aggregate consisting of tightly packed beta sheets. Okay. So protein amyloid aggregation has been recognized as a major cause of several uh, important diseases such as Alzheimer's disease, Parkinson's disease, type 2 or non insulin dependent diabetes, and the transmissible spongiform encephalopathies. About 17 different proteins have been found to form amyloid in vivo. Amyloid fibrils formed from these proteins share some common morphological features, but these proteins do not have a conserved sequence or native structural motif. Okay, so protein amyloid fibrils often found to have a beta sheet structure regardless of their sequence, leading some to believe that it is the molecule's misfolding that leads to aggregation. Okay. Now, how would the chaperones do? Chaperones are proteins. How proteins get help in folding? So, if you see, if you can read more about the GROW EL system. So, in eukaryotes, the proteins HP, heat shock protein 60 and heat shock protein 10 are structurally and functionally nearly identical to GROW EL and GROW ES respectively. Okay, so what is the chaperonin steps of chaperonin action? So an unfolded polypeptide enters the cylinder from one end, right? The cap attaches, causing the cylinder to change shape in such a way that it creates a hydrophilic environment for the folding of the polypeptide. Then the cap comes off and the properly folded protein is released. In general, it is actually quite poorly understood how it works, whether it is active in terms of interactions between chaperonins or inert, where chaperonins simply make an environment conducive to folding is not known. It is, however, required for the proper folding of many proteins. Okay, chaperones with ATP environmental conditions suitable for proper folding. So binding of a 7 ATP molecules to the GROW-EL ring triggers conformational change that results in slight twists and tilt in subunits and in exposure of the hydrophobic patches that interact with and help to unfold the misfolded proteins. So the destruction and building of the secondary and tertiary structure of proteins, denaturation and folding of proteins. So proteins are synthesized on ribosomes as linear polypeptides. We know about this. As they synthesize, they assume secondary and tertiary structure, and activity of the proteins depend on the integrity of its final tertiary structure, also referred to as the native form. The native form of protein is not very stable. It is vulnerable to change by heat, high salt, reducing agents, detergents, organic solvents, etc. All these treatments destabilize the weak interactions responsible for the higher order structures. So the destruction of the higher order structures leads to the loss of activity of a protein, and this process is called the denaturation or unfolding of a protein. Now, denaturation may not mean complete unfolding of a protein, it might still be a folded structure but in random conformation. So, denaturation is cooperative, that it changes in one part of protein accelerate the unfolding of the other part. Now, some proteins are resistant to denaturation by heat. The primary structure of these proteins are not very different from those from normal bacterium. So, it remains a biochemical puzzle to explain the stability of these proteins actually. Okay, so we discussed so how the folding of polypeptides is rapid in a stepwise fashion. As I said, proteins synthesized and folded in different conformation, and the one of which matching with native conformation is selected. Okay, is not possible. So proteins keep folding in the correct conformation as it is synthesized in a stepwise fashion. So initially, the unfolded peptide has highest randomness, and as it keeps assuming more folded structure, then the entropy keeps on decreasing. Okay, this is how the folding happens. Basically, it starts one and then the steps start going on. So not all proteins fold spontaneously as they are synthesized in the cells. Folding of many proteins is facilitated by a specialized class of proteins called chaperones, as we said, like the heat shock type proteins, chaperonin, GROW ES, other enzymes involved in protein folding such as protein disulfide isomerase (PDI), peptide prolyl cis trans isomerase (PPI), all of these. Okay, we'll, this is some of the diseases that are caused by misfolding of proteins. I'm not getting into the Okay. So basically, molecular chaperones are a large group of unrelated proteins families whose role is to stabilize, right? Unfolded proteins, unfold them for translocation across membranes or for degradation and to assist in their correct folding and assembly. Now, molecular chaperones interact with unfolded or partially folded protein subunits, for example, nascent chains emerging from the ribosome or extended chains being translocated across subcellular membrane, and they stabilize non-native conformation and facilitate correct folding of proper protein subunits. They do not interact with native proteins, nor do they form part of the final folded structures. Some chaperones are non-specific and interact with a wide variety of polypeptide chains, but others are restricted to specific targets. They often couple ATP binding hydrolysis to the folding process and is essential for viability. Their expression is often increased by cellular stress. So, the main role is to prevent inappropriate association or aggregation of exposed hydrophobic surfaces and direct their substrates into productive folding, transport, or degradation pathways. Right? So, examples of molecular chaperones are heat shock proteins, as I said, catalysts for folding like PDI, protein disulfide isomerase, peptidyl prolyl cis trans isomerase PPI, nucleoplasmin, which helps in nucleosome assembly, and all of this. Okay, not go into these details. As I said, protein folds into single energetically favorable conformation specified by its amino acid sequence, and they can fold into alternative 3D structure due to mutations or inappropriate covalent modifications, right? So, correctly, newly synthesized protein can correctly fold without any help, correctly fold with the help of molecular chaperone, or incompletely fold form digested by proteasome, or if they stay they can even form protein aggregates which are very harmful. So accumulation of misfolded protein or proteolytic fragments results into few degenerative diseases and characterization by presence of insoluble protein plaques in organs such as brain and liver. Okay, so we have learned about all of these. Okay, so coming back to the question that we started with, chaperones are examples of ribonucleases. It's false. Okay. So anybody has any question regarding this? I will wait for 1-2 minutes, then we'll move on to the next question for today. Okay, I'll take a few minutes break. You can let me know if you need more, if you have any queries regarding this question, then we'll move on to the next question. Okay, so moving on to the next question for today. The alpha helix sequence in protein GB1 has the sorry. The alpha helix sequence in protein GB1 has the sequence AATAEKVFKQYANDN. How many full turns are in this alpha helix? So we know the options 4.8, 5.4, 3.6, and 4.2. So which one do you think is the correct answer? Anybody? We discussed in one of the last questions how many residues are in alpha helix one turn. So based on that knowledge, you should be able to answer this question. Anybody? Anybody? We discussed about the concept of a full turn in an alpha helix in one of the last questions. Okay, so the answer will be 4.2. Why? So if we think about the concept of a full turn in an alpha helix there are 3.6 residues per turn, right? We have discussed about the different levels of protein structures. For example, we have the primary structure, which is a sequence of chain or of amino acids. Then we have the secondary protein structure, which occurs when the sequence of amino acids are linked by hydrogen bonds. Then you have the tertiary protein structure, which occurs when certain attractions are present between alpha helices and pleated sheets. Then we have the quaternary protein structure, which is a protein consisting of more than one amino acid chain. Now, primary structure is defined as a sequence of amino acids in protein. Understanding the primary structure of proteins is important. So, because many genetic diseases result in proteins with abnormal amino acid sequence which cause error folding and loss or impairment of normal function. Now, in proteins, amino acids are joined covalently by peptide bonds, which are amide linkages between the alpha carboxyl group of one amino acid and the alpha amino group of another. For example, the formation of the peptide bond between glycine and alanine. Peptide bonds are broken by prolonged exposure to strong acid and base at high temperature to hydrolysis, and these bond non-enzymatically. Okay. So there's a free amino end is written to the left and free carboxyl end is written to the right. Therefore, all amino sequences are read from the N to C terminal end of the peptide. So polypeptide are linkages of many amino acids through peptide bond results in an unbranched chain. Now residue or moiety, each component amino acid in a polypeptide. When a polypeptide is named, all residues have their names changed with exception to the C terminal amino acid, right? What are the characteristics of a peptide bond? Partial double bond character, that is, it's shorter than single bond. Rigid and planar, this prevents free rotation around the bond between carbonyl and nitrogen of peptide bond. Trans configuration because of steric interference of the R groups when it, the cis position and uncharged but polar. Determination of the amino acids composition of a polypeptide. So the determining of primary structure of polypeptide is to identify and quantitate its constituent amino acids by hydrolyzing with a strong acid at high temperature for 24 hours. So to release the individual amino acids which can be separated by cation exchange chromatography. So cation exchange chromatography is nothing but a mixture of amino acids bind to the column that contain a resin which are negatively charged group with different affinities depending on their charges and hydrophobicity. So separate amino acids contained in the eluate with solutions of increasing ionic strength and pH are quantitated by heating them with anhydrine, which is a reagent that forms a purple compound with most amino acids. So the amount of amino acids is determined spectrophotometrically by measuring the amount of light absorbed by anhydrine derivatives. The analysis described is performed using an amino acid analyzer. So we'll not go into much details. Secondary structure. So it's defined as regular arrangements of amino acids that are located near to each other in the linear sequence. The alpha helix, beta sheet, and beta band are examples of secondary structure. Alpha helix is a spiral structure consisting of a tightly packed coiled polypeptide backbone core with the side chains of the component amino acids extending outward from the central axis to avoid interfering sterically with each other. Alpha helix. Okay. Then we have the very diverse group of proteins contain alpha helix. The keratins are a family of closely fibrous proteins, major component of tissue such as hair and skin. Keratin and myoglobin is globular flexible molecule whose structure is approximately 8% alpha helical and their rigidity is determined by the number of disulfide bonds between the constituent polypeptide chains. Hydrogen bonds and alpha helix is stabilized by extensive hydrogen bonding between the peptide bond, carbonyl oxygens, and amide hydrogen that are part of the polypeptide backbone. The first and the last peptide bond component are linked to each other through hydrogen bonds. Hydrogen bonds are weak, but they collectively serve to stabilize the helix. Now, amino acid per turn. Each turn of an alpha helix contains 3.6 amino acids. Thus, amino acid residues spaced 3 or 4 apart in the primary structure are close together when folded in the alpha helix. Okay, so there are 3.6 residues per turn on average. So one full turn is 3.6 amino acid residues. Now the sequence that was listed, there are total 15 residues. So number of turns will be 15 by 3.6, which is 4.17, or 4 point, which is the closest to the 4.2 in the options range. So basically the alpha helix will have 4 full turns plus a partial turn at the end. Okay. So what are the amino acids that disrupt an alpha helix? Like proline disrupts an alpha helix. Why? Because its amino group is not geometrically compatible with the right-handed spiral of the alpha helix. Then we have the charged amino acids like glutamate, aspartate, histidine, lysine, and arginine. They disrupt the helix by forming ionic bonds or by electrostatically repelling each other. Amino acid bulky side chains such as tryptophan can also disrupt an alpha helix, and a large number of amino acids like valine or isoleucine that branch at the beta carbon can interfere with formation of the alpha helix. Then you have the beta sheet is a second form of regular secondary structure in proteins. They are less common than the alpha helix. The structure of beta sheet appear pleated. Beta sheet strands are often visualized as broad arrows. So beta sheets are composed of two or more peptide chains. The hydrogen bond in beta sheets are perpendicular to the polypeptide backbone, and parallel and antiparallel sheets. Beta sheet can be formed from two or more separate polypeptide chains that are arranged either antiparallel or parallel. So antiparallel beta sheet polypeptide chains are arranged to each other with the N-terminal and C-terminal ends of the beta strands. Polypeptide chains are arranged to each other with all the N-terminal of the beta strands together. The hydrogen bonds are formed between the polypeptide backbones of separate polypeptide chain. A beta sheet can also be formed by single polypeptide chain folding back on itself. In this case, the hydrogen bonds are intra-chain bonds. We'll not go into much details on this. We have talked about motifs and domains. Near the tertiary structure of protein, which is basically the total 3D conformation of an entire polypeptide chain, including alpha helices, beta sheets, and any other loops, turns, or bends. Structure of globular proteins in aqueous solution is compact with high density of the atoms in the core of the molecule. Hydrophobic side chains are buried in the interior, whereas hydrophilic groups are found on the surface of the molecule. Okay, so disulfide bonds is a covalent linkage formed from the sulfhydryl group of each of two cysteine residues to produce a cysteine residue. A disulfide bond contributes to the stability of the three-dimensional shape of protein molecule and prevents them from denaturation in the external environment. Now, hydrophobic interaction amino acids with nonpolar side chains tend to be located in the interior of the polypeptide molecule, where they associate with other hydrophobic amino acids. In contrast, amino acids with polar tend to be located on the surface in contact with the polar solvent. Amino acid side chains containing oxygen or nitrogen bound hydrogen can form hydrogen bonds with electron rich atoms such as oxygen of a carbonyl or carboxyl group of a peptide bond. Formation of hydrogen bonds enhances the solubility of the protein. Negatively charged groups such as the carboxyl group in the side chain can interact with positively charged groups such as the amino group in the side chain of polypeptide. So as we discussed in the last question, a special group of proteins called chaperones are required for the proper folding of many species of proteins, and also known as the heat shock proteins interact with the polypeptide at various stages during the folding process. Some chaperones are important in keeping the protein unfolded until its synthesis is finished, or act as catalysts by increasing the rates of the final stages in the folding process. They protect proteins as they fold so that their vulnerable exposed regions do not become tangled in unproductive encounters. Then we have the quaternary structure of protein, which is the arrangement of multiple folded protein or coiling protein complexes in a multi-subunit complex. We will not go into much this. We have already talked about all of this. coming back to the question that we started with today about alpha helix and the number of turns. So each alpha helix has 3.6 amino acids in each full turn, right? So with a sequence of 15 amino acids, as we can see over here, the alpha helix will have 4.2 turns, 4 full turns, and then last a little bit bent at the last. Okay, so this was the last question for today. If you have any queries, you can ask me now, and then we'll end the session for today. Okay, so if there are no more questions, we can end the session here today. We'll meet again next Monday, same time, 6 to 8 p.m. Thank you so much.